On to Aptamers. Thank you all for joining us, and thanks for the support. So the team here is myself, Bert Semler, and Mike Bachmeyer, who keep me in check, my imagination, and all that. If you sort of think about the coronavirus replication, here's a list of the key steps that you might want to interfere with or use for, so either for therapy or diagnostics. And so we're focusing on the S-glycoprotein because that's sort of the first point of contact. That's what touches the receptor and helps in release of the viral RNA in the host cell. And so we have two parallel aims, two parallel experiments initially, which is basically doing an in vitro selection for aptamers that either block directly the receptor binding domain of the S-glycoprotein, or actually switch conformation that can be used directly in diagnostic tests. And so this is very much equivalent to antibodies. I'll show you a bit more about that. So just to bring you into the 3D, perhaps projected 3D space. Here what you see on the left is a recent cryo-EM structure of the current SARS spike protein. The receptor binding domain is in green here, and they modeled it sort of in two different conformations. When they looked at, in a different recent cryo-EM structure of the ACE2 receptor for the virus, and overlaid the receptor binding domains from the old SARS and the new SARS, you can see that they're very much similar. So that's this green and golden structures docked on, but the details are different for the two. And then other papers that have come out recently, including some crystal structures, all sort of give the same overall shape of the interaction, and all the details appear to be a little off. So either these are model biased, or there's actually some dynamics going on. We won't know for a little while, but nonetheless, the idea here is that the spike protein docks onto the receptor in a very similar way between the original SARS and the COVID-19 SARS virus, coronavirus. And importantly, and this is something that has come up in our reviews, both the receptor and the spike proteins are glycoproteins, but the glycosylation sites are sort of away from the interface. So if this is the interface, you can see that these are the glycosylation sites. They're nearby, but they're not at the interface. And so that's all protein-protein interactions. And that perhaps helps. Perhaps correlated with some of this is the fact that if, here's a recent crystal structure of this is a model. So the crystal structure was between this antibody, the CR3022, and the receptor binding, just the receptor binding domain, and then modeled on as the interaction with the ACE2 receptor. And so you can see that they're in a similar place. And then on the bottom here is an overlay of various antibodies against the receptor binding domain from the old SARS coronavirus. And so these antibodies are all sort of in the same area. There appears to be a sort of a preferred interface. And so to us, this means that would probably be the best area to target with other molecules. So in our case, with aptamers. The assay that Bert Semler's lab and Mike Bachmeyer's lab have developed is relatively simple. There's an effect that if you overexpress just the spike protein, so nothing else from the coronavirus, whether the old or new, the first thing that you observe, and this is in HeLa cells, so not epithelial, lung epithelial cells or anything like that, you see immediately cytopathic effects. And you can see it here a little better. You can see oligonucleated cells. And so the basic interpretation of that is that the spike protein is doing what it's doing on a virus. It's trying to merge these two cells. They're just expressing it, right? So this is just from expression vectors. And again, oh, sorry, this is a little shifted. So the top one is the old SARS, the bottom one is the new SARS, and the red is a stain for an epitope on the expressed spike protein. So you can see that the, in this case, trinucleated cell is expressing the spike protein. And so this is an easy assay. You know, you sort of look, see what you can do and whether you can interrupt this just in culture. And so we're trying to develop aptamers, and you can think of them essentially as antibodies, but they're developed completely in vitro, and you can synthesize them because they're just pieces of RNA or DNA. So in our case, it'll be single-stranded DNA, and here's just one scheme that we're using where you take a rather large...
This is the 5-minute mark?
Good. Yeah, I have about 30 slides to go. And so, so we start with a very large 10 to the 14, 10 to the 15 different sequences and select the ones that can do what you ask them to do, in this case, binding of the targets. And so the shapes, this is just examples for aptamers for thrombin and some plasmodium enzyme. The interaction is very similar to antibodies, but they're a lot smaller, right? And so we've already gone through 9 rounds of selection. The initial libraries for the aptamers are being sequenced as we speak. So we're making good progress. Hopefully we'll see what comes out, and then we'll take it into the culture.
Okay, that's great. We do have time for questions. So I can start with one. With the antibodies against the original SARS-1, what is the evidence that those were neutralizing?
I don't know. Mike, do you know?
Sorry, I'm muted. I don't know the source of your antibodies. I would assume if they were given to you as neutralizing antibodies that they'd been tested on SARS-1. I've just reviewed a paper for Journal of Infectious Disease, which shows that antibodies which neutralize SARS-1 also react with SARS-2 and will neutralize to a lower extent SARS-2. So there seems to be enough basis for that to assume that that's correct.
Thank you.
There is another issue, however, which I was going to suggest to you, Andre, and that is, you know, if you go far enough back in my curriculum vitae on coronaviruses, you'll find that there's an antibody which we described in 1982, which blocks fusion. And that antibody is also a neutralizing antibody. When we tried to select variants that were resistant to that antibody, the virus was very resistant to allowing us to select mutants that were resistant. They were dead or they were fully functional. So it's suggesting that it's a very important site. And that is a site that lies between cleavage site number 1 and the TMPSRSS2 site, which is the secondary site that's so critical. So that may be another target to consider. It's not in the RBD, but it's in a functional domain in the glycoprotein.
Yeah, we would have to start over for that one.
Yeah.
But this Dr. Perlman, I have a question also. These are really compelling data with the fusion and the multinucleate cells there. Do you have a sense how much of this actually happens in the lungs in infected tissue?
Yeah, do we have any pathologists here?
I have experience with that with SARS-1. Those cells are evident in the lung. CDC did the pathology very early and showed multinucleate giant cells in the lung and also in sputum. They could recover the multinucleate giant cells of the same morphology. That's not uncommon in lung disease. You find it in measles as well, so it's not so unusual.
What effect do you think it has on the outcome of disease, if any, severity of disease?
It's part of the destruction of the respiratory system. It's one of those things that's very hard to recover from. You know, these cells are recruited into multinucleate syncytia that are taken over by the virus. And it's hard to regenerate when that's going on at the same time. It can wipe out 100, 200 cells at a time.